Hello guys, so this is the second video of the pancreas, and today we'll be doing the histology part and the functions of the pancreas, the structures which are present inside the pancreas. It's the alpha cells, the beta cells. We all know that the pancreas is responsible for reducing the level of glucose, but we don't actually know that where the cells are present that release the insulin into the GIT to reduce the glucose level. So let's get started. We'll do the histology of the pancreas. Now I will be making the diagram. Let's write the heading. This is histology, and this is the I will be making the diagram now. It's super simple. The whole GIT, not the whole GIT, but most I will be drawing just to demonstrate the uh, the function of the liver, how the liver is present and how it releases the various enzymes into the circulation. We all know that this is the unkinated process I have discussed in the previous video. Now we will be making the ducts of the pancreas. This is the major pancreatic duct. It is coming from the tail and this is the major papilla of the duodenum, and this here is the bile duct which is coming from the common bile duct, which is coming from the in the back of this pyloric duodenal junction, and this is coming from the we all know that bile duct comes from the liver. So I will be drawing liver over here. This is the liver. Okay guys, we know that this is the left hepatic duct and this is the right hepatic duct, and this is the gallbladder, and this is the cystic duct, and this is the common bile duct, and the common bile duct joins here and forms the major pancreatico-hepatic duct. So this name is important, major hepatopancreatic duct. Let's write over here, major pancreatico-hepatic duct. And there are various interlobular ducts which are the branches of this major pancreatic duct. I will just show you the branches now. So we all know that here is the accessory duct. So this major pancreatico-hepatic duct is also known as duct of Wilson. And now I will be drawing the branches. Various branches over here. So these are the various branches. Interlobular ducts, these are known as interlobular ducts. I will be finishing them. So in these branches there are present the acini are connected to these branches. So these are the acini. We all know that the pancreas has dual function, endocrine as well as exocrine. So as I think you remember that acini cells release digestive enzymes into this GIT. So the acini cells are exocrine cells because they have well defined ducts present. So in between these there will be present, can you recall what are these? So these green cells are called islets of Langerhans. These are the responsible for the endocrine portion. I will enlarge this diagram later. First we will do the exocrine portion, and before that, after this I will like to draw the histology diagram of this interlobular duct, the connective tissue. We know that the connective tissue is present in the pancreas. The connective tissue has various septa which divide the parenchyma of the cell of the gland into various lobules. So we can write here, there is connective tissue present. Let me see, video is running smoothly. Yes, it is. Which divide the septa. No, it has the septa. Connective tissue has a septa which divides the parenchyma of the gland into various lobules. So here is the sphincter of Oddi, which is very important to remember. Here is the minor papilla, and the region where the sphincter of Oddi is present, it is called the major papilla. And one more thing, the location, I would love to tell the location of the enterokinase. This is present over here. This is the location of enterokinase. Enterokinase has a special function, I will discuss it later. Then after that we will be drawing the diagram of the after that we will be drawing diagram of the pancreas, which is showing the which we will be shown in the slides. So we should know that how to identify that diagram. So I will draw the outline. That's the rough outline. What are these? These are pancreatic acini. This is the connective tissue which is dividing the gland into the lobule, as I have already written before. So in between that there will be present islets of Langerhans. So they will be like this. Alpha, beta, alpha, beta, alpha, beta, delta. These cells would be present here, and the acini cells can be detected at the periphery. There would be lots of pyramidal cells present. They would be staining different color. You can easily identify these cells. And more importantly, this is the intralobular duct. There is the concept of intralobular duct and interlobular duct. Okay, so these nuclei will be present at the base in the interlobular duct. So I will just explain you what are these and what are these for. Like this is the tree. This is the one branch. This is the one branch of the tree. No. Here is the, imagine here is the one branch of the tree. So this is the interlobular duct of the tree, and this branch again forms various branches. So these various branches are intralobular ducts. So in this way you will be able to imagine that how these are present. So these are the islets of Langerhans. So various acini will be present here that will be draining their exocrine portion, that is digestive enzymes, into these intralobular ducts, and these intralobular ducts will be draining their all the secretion of the, collecting all the secretions of the acini and draining into the interlobular duct. This is the interlobular duct. Interlobular duct so this stem of the main stem is the major pancreatic duct. So this stem is the major pancreatic duct. You can easily demonstrate by this what I want to convey you. So this was the, so you can imagine, like I have not made the whole diagram, it would be consuming a lot of time. So here all will be this, these all will be present, and like here alpha beta alpha beta alpha beta. So these cells would be elongated cells. You will able to recognize in the slides, and these will be the rounded, the acini cells. So let's move ahead. Now we will be doing the exocrine portion. Exocrine portion, as you all know, the acini play an important role in the exocrine part. So let's start with the exocrine part. First I will make the diagram of acini, then I will make the diagram of the single acinar cell, that is pyramidal cell. As you can see, these intercalated ducts, this is the intercalated duct. As you can see that I have made it quite inside. It is not a mistake. This is the property of these intercalated duct. I will explain you. These are various pyramidal cells present in the acini. These all cells so these are the zymogen granules, zymogen vesicles present. Golgi, this is the Golgi, where the packing occurs of the pro-enzymes. I will explain it all. So this is the Golgi, and there is the nucleus present. This is the nucleus. In all these, nucleus is present, and now let's draw the intercalated ducts properly. So the intercalated ducts which are present inside the lumen of the acini, these are called centri acini. These are called centri acinar cells. Centri acinar cells. Okay, and there is a covering also, and they have various receptors for acetylcholine, CCK, cholecystokinin, and acetylcholine. Intercalated ducts also have the receptors for secretin and acetylcholine. So intercalated ducts extend inside the lumen of acini cells. These extended cells are called centrally acini cells. Exocrine portion consists of acini and draining duct system. These acini are made up of pyramidal cells. Pyramidal cells release digestive enzymes into the lumen. Within the lumen there is the formation of intercalated ducts. Intercalated ducts secrete bicarbonate ions. So you will be thinking that if you have an active brain, you will be thinking why there is need to secrete the bicarbonate ion, that is alkaline. The pancreas can simply release the enzymes, the digestive enzymes for the protein digestion. It releases we know chymotrypsin, trypsin and all that. It can simply release, but the alkaline part has a lot of important function, because the food which is coming from the stomach, it is very acidic. As you know that the stomach has acidic pH around 1.6. so the food is coming from there, and it is entering into the food is coming from there and entering into the duodenum part. And for the breakdown of the proteins, it needs a basic environment, so this is provided by the intercalated ducts. So the intercalated ducts secrete the bicarbonate ions. If the cells of intercalated ducts are stimulated by acetylcholine or secretin, they will release the bicarbonate ions. If the acetylcholine stimulate the acini cells, it will release the digestive enzymes. You can now think that acetylcholine has important function in digestive enzyme synthesis and in alkaline medium maintenance. So it stimulates the ductal cells and stimulate the release of bicarbonate, as well as it stimulates acini cells and increase the release of enzyme. But CCK and secretin are specific enzymes. CCK stimulate the acini to secrete the digestive enzyme. CCK specifically stimulate the acini to secrete digestive enzymes, and secretin specifically stimulate the intercalated ducts to release the bicarbonate ions. When food comes from gastric environment, it is very acidic. To neutralize the acidic environment, the pancreas releases bicarbonate, then pancreatic enzymes can work. As I have already told you, this all was I was reading the theoretical part, I have explained you before. So now let's draw the diagram of the single pyramidal cell of the acini. So as I have mentioned before, there will be the presence of Golgi bodies. These are the Golgi bodies. There will be present nucleus and RER, rough endoplasmic reticulum, ribosomes and ER. There would be present free ribosomes also. So this is the nucleus. This is the RER. This is the ribosome. Okay. So the single acini cell is protein synthesizing and secreting cells. So the peptidases come over here, like this is the peptidases. So it needs the activation. So the Golgi apparatus do this function. These proenzymes are processed within the Golgi and packed into secretory or zymogen vesicle These vesicles are rich in enzymes. Enzymes move to luminal sites. When these vesicles are rich in enzymes, this enzyme move to luminal sites. So this is the function of the Golgi. This is the Golgi. Then comes the which enzymes. Which enzymes are that? This is the important question. Can come. Which enzymes? I'll show in the next page. Endopeptidases, exopeptidases. Um, First we will write the endopeptidases. You should remember that it is chiefly important for the protein synthesis, protein breakdown, sorry. So it breaks the peptide chain from within. It breaks the peptide chain from the center. Exopeptidase, it breaks the peptide chain from the terminal ends. From terminal ends. The example is chymotrypsin. I'm sorry. Chymotrypsinogen and trypsinogen. After that, I will tell you the importance of enterokinase. It is related with the chymotrypsinogen and trypsinogen. So exopeptidases break the peptide chain from the terminal ends. Procarboxypeptidase proaminopeptidase. So these are the examples of the exopeptidases, and they are also present for the lipid breakdown. So the lipases and pancreatic cholesterol esterases. So these are the enzymes for the lipids, and there are present amylase, which is also present in the mouth, and deoxyribonucleases are also present. Deoxyribonucleases and ribonuclease is also present. Okay, so these are the enzymes present. Then we will I will tell you the function of enterokinase, which was present on the, I have made the red stars on the GIT. So the function of enterokinase is responsible for activation of trypsinogen into trypsin. Activation of trypsinogen into trypsin, and this trypsin is responsible for the activation of other proenzymes. Okay, so this was all about the exocrine portion. Now we will jump to the endocrine portion. Let's go. For endocrine portion, I would like to draw the flow chart. So endocrine, first of all, this is the diagram of the, as you can visualize, this is the diagram of the this. Here are beta cells present and alpha cells present. Beta cells are more present. So these are alpha, beta, beta, beta. We will try to, and this is somatostatin, alpha, alpha. this beta, 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 beta. So this is the structure of the islets of Langerhans. So now the cells which are present in islets of Langerhans, I will like to draw the flow chart, which will make you easily, which will make your learning easier. So let's draw it. Types of cells, percentage in islets of Langerhans, secretion function and its function. Okay. So the first one is the alpha, 20%. Secretion, we all know that alpha releases glucagon. So and its function is rise in blood glucose level. Then beta, most are beta, 70 to 75. Secretion insulin, it has various clinical practice, we all know. Decrease in blood glucose level. Then comes the delta, we can do delta, it's 5 to 8%, I can remember, and it is somatostatin, somatostatin. So the function of somatostatin is that if the alpha cells and beta cells are produced too much, it inhibits both alpha and beta cells if raised beyond their level. So this is the checker. We all need a checker on our life. So this is a checker on alpha and beta, so that no abnormality can be produced. So this is a plasma protein, 1%, pancreatic peptide, inhibition of the exocrine secretion of the pancreas. So this is the time checker. So this is the checker for the exocrine secretion of the pancreas. Inhibition of exocrine secretion of pancreas. Okay. So this was all about the histology and the functions, the structures which are present, their functions. Now in the next video, let's see, we'll most probably do the development and then the abnormalities of the clinical anatomy of the pancreas. Thank you for watching. Have a good day.